Let us discuss about constitutive secreted proteins. An example of constitutive secreted protein is serum albumin. Serum albumin is secreted by the continuous pathway. Proalbumin is the precursor of serum albumin. Proalbumin is a proprotein. Like all other proteins, proalbumin has an amino terminus and a carboxyl terminus. The proalbumin has a dibasic recognition sequence. This dibasic recognition sequence has two consecutive arginine amino acids or lysine arginine. The enzyme furin endoprotease cleaves the proalbumin at a site C-terminal to the dibasic recognition sequence containing two consecutive arginine amino acid or lysine arginine. Due to this cleavage, the dibasic recognition sequence separates from the rest of the sequence of proalbumin. The remaining sequence of proalbumin after separation of dibasic recognition sequence is called as albumin. Furin endoprotease is found in all mammalian cells. Now let us discuss about regulated secreted proteins. An example of regulated secreted protein is insulin. Proinsulin is the precursor of insulin. Proinsulin is a proprotein. Like all other proteins, proinsulin has an amino terminus and a carboxyl terminus. The proinsulin has three polypeptide chains like polypeptide chain B, polypeptide chain C, and polypeptide chain A. Polypeptide chain B is present at the amino terminus of proinsulin. Polypeptide chain A is present at the carboxyl terminus of proinsulin. In between polypeptide chain B and polypeptide chain A, polypeptide chain C is present. At the amino terminus of polypeptide chain C, two consecutive arginine amino acids are present. At the carboxyl terminus of the polypeptide chain C, lysine and arginine amino acids are present. Polypeptide chain A and polypeptide chain B of proinsulin are joined by two disulfide bonds. Polypeptide chain A has one intrachain disulfide bond. PC3 endoprotease proteolytically cleaves at a site C-terminal to the two consecutive arginine amino acids. PC2 endoprotease proteolytically cleaves at a site C-terminal to the lysine and arginine amino acids of polypeptide chain C of proinsulin. Due to the action of PC3 endoprotease and PC2 endoprotease, part of C polypeptide chain having two consecutive arginine amino acids remain attached to the C terminus of polypeptide chain B with the formation of a part of polypeptide chain C having lysine and arginine amino acids. The part of polypeptide chain C having lysine and arginine is degraded. The carboxypeptidase then sequentially acts on two consecutive arginine amino acids in the part of polypeptide chain C of proinsulin. As a result, a mature insulin is formed with the removal of two arginine amino acids. Relative molecular mass of mature insulin is 5800. Now some important points. Proteolytic cleavage of proinsulin occurs in vesicle after they move away from the trans-Golgi network. PC2 endoprotease, PC3 endoprotease, and carboxypeptidase are present in regulated secretory vesicles. The examples of proproteins that undergo proteolytic processing to produce mature active proteins are soluble lysosomal enzyme, many membrane proteins such as hemagglutinin or HA, and secreted proteins such as serum albumin, insulin, glucagon, the yeast alpha mating factor. Please like, subscribe and share.